Carrier-mediated transport. When we have carriers involved, it's going to be very specific. Okay. Specificity. Whenever you see a facilitator, you always should think specificity. Okay. Now, I'm going to say specificity, and then I'm going to say, well, something else could get in there and dock with that receptor. So it can compete. There can be some competition. But when I'm saying specific, I'm meaning specific shape. Okay. Different ligands have different shapes. Some ligands have very similar shapes. Certain drugs, okay, have similar shapes to various different receptors within the brain. You have some medications involving the treatment of brain disorders that have the exact same shape in the digestive tract. So medication that you're taking for depression and whatnot goes and affects the digestive tract as well at the same time. And it's going to, in one region, it's going to cause some activity to downplay. And in another region, it's going to cause some activity to increase. So in that situation, it's the same shape. Okay? So when I say they're specific, yes, they are specific, but it's a specific shape. Okay. And as long as it's the same shape, it can dock to that specific receptor. So there are some downfalls to this idea of carrier-mediated transport and it being specific. A number of various different things have specificity. And again, these are all very large, very polar things. All right. They have charge to them. They don't diffuse through the lipid membrane very well at all. Okay. They're repelled. So we're going to have a lot of things that are going to help bring that across. There is a limit to which we can facilitate and move things across through a carrier. Carriers are going to be very limited by just the sheer number that are present. If we have a lot of carriers, I have the potential to move a lot of substances across. If I have very few protein carriers, I can only bring very few things across. Can the body change that? Hell yes. Okay. In different situations, the body can insert more of those carriers onto the surface. In some regions, it can take them away in some instances. Okay. And we'll talk a little bit about upregulation and downregulation because it's a phenomenon that we see quite a bit, especially involving hormones and whatnot within the body. Some of these carriers can transport more than one thing, and transport rates. Whoops. Okay. So some proteins can transport more than one thing, and we'll talk about some of those specific ones here in a few seconds. Sometimes they can compete. Sometimes they don't compete. So there is a competition involved. I will say this in terms of minerals that you consume in your diet. There are a number of minerals that share transport mechanisms like this. And if you consume too much of one type of mineral, it can affect other minerals as well. And you can become pretty deficient in minerals in that regard. So, having a varied diet really helps to limit that. But if you're somebody who supplements or over supplements, you can flood the system with too much of one specific type of mineral, and that would prevent other minerals from coming across into the digestive tract. So there is some competition that exists, and we'll hint at some of that phenomena when we get into the digestive system. But again, this ability to bring stuff across, a lot of stuff across quickly, or to bring a few things across quickly, again, is dependent upon the number of carriers that are present. We can talk about this in terms of saturation of those carriers. There is a limit, and that limit is the transport maximum here. And here's just a nice little fancy pants graph to kind of show that off, all right? You can see the rate of transport increases with an increase in the concentration. When the body increases its concentrations, the body will upregulate and insert a number of protein carriers in its surface, in its plasma membrane, and that will help to increase this transport maximum to occur there. It gets to a point where it gets saturated, and then it doesn't fall off. Okay, you don't see this. You don't see reach saturation and then all of a sudden, bam, you don't see that at all. But what do you see? You see it leveling off for the most part. Okay, you see it leveling off for the most part. And that's what happens when you reach transport maximum. When you saturated all of those protein channels, okay, it doesn't fall off. It doesn't drop off. It levels out. Okay. And if there's going to be something that's very easily missed on an exam, that is generally the concept. Okay. It doesn't fall off. It doesn't drop off. All right. You saturate everything and now it stagnates. Okay. Stuff is still moving across. And as soon as you move something across, now that carrier is open. So something can dock into that carrier and it can come across. It just now it slows down tremendously to the point where it levels off. Okay. Still transportation going. It just doesn't go any faster because you've saturated every single protein carrier. One of our carrier-mediated mechanisms is facilitated diffusion. Again, when we have facilitated diffusion, it's going from high to low concentration. It's just big or charged. So we need to have a specific thing, carrier, to bring it across. So facilitated diffusion is, even though it uses a carrier, there is no ATP expended because, again, we're going from high to low concentration. And here is a fantastic general example showing that phenomenon. Okay. There's glucose. It's big. Can't easily come across. And we have our specific carrier protein to bring it across. What is the name of that specific carrier? It is called a glucose transporter. And we use the term GLUT. Okay. Glucose is the best example when it comes to facilitated diffusion. There are, I can't remember off the top of my head, 18 or 20 different glucose transporters all over the body. Medication is tailored to those specific glucose transporters. We have glucose transporters in the brain, in the central nervous system. We have glucose transporters within, obviously, the pancreas, the liver. One's just for the neurons. One's even for adipose and skeletal muscles. Okay, so we can target medication for a specific glucose transporter. So we don't just turn them all on or shut them all down. Okay. Depending upon the situation, your body, our bodies, will put these GLUT transporters into the plasma membrane of a cell when the need arises. Okay. We have a number of receptors in the surface of the plasma membrane that will be there and that will dock with insulin. Insulin is a hormone, right? Secreted by the pancreas. It will upregulate, okay, cause the insertion of all of those glucose transporters into the surface of the plasma membrane. When sugar levels are high within the blood, insulin helps to facilitate that movement of glucose into the cells. And what's glucose going to do? Obviously, it's involved with making ATP. We can saturate those, okay? So if you consume too much carbohydrates, right, you get all that excess sugar within the blood, and that can ultimately be a bad thing. For people who can't release insulin or have insulin imbalance, well, especially if they're a type 1 diabetic and don't have insulin secretion, glucose can't get into the cell very effectively. Right? There's not a lot of protein receptors, not a lot of glucose transporters on the surface of a plasma membrane in general. And so when all of a sudden a type 1 diabetic eats all that carbs and gets all that glucose in there, they don't have a way to upregulate or insert all of those glucose transporters into the surface of their plasma membrane. Okay? So it can't bring it across. So I have all this sugar within the blood, and we convert some of that into fat. Some, can get into the urine, and that's telling you have some kidney damage. So how in the heck does somebody who has type 1 diabetic get anything in their cells? Glucose was the primary thing, right, to provide energy. So what do they switch over to? Fats. So someone who's type 1 diabetic, what's their typical appearance? Rail thin. They have a sugar imbalance. They can't get sugar into those cells to provide it with energy. So what are they going to draw upon? Those triglycerides, those fat stores. And they can get those easily across. Sometimes we need a carrier to help things go against the concentration gradient. When we go against the concentration gradient, we go from an area of high concentration to an area, or from an area of low concentration, to an area of high concentration. These mechanisms always require ATP. To go up the slide requires effort, a lot of it. Going down the slide doesn't require any effort, going from high to low. When you go backwards up the slide, that takes effort, and that requires ATP. We call these pumps sodium-potassium pump, okay? Calcium pumps. These are going to be things that require energy, that ATP. When we think of active transport, this is what you think about right here. Okay? Biology 101, hell, even in, I talk about it in 109. But in Biology 101, when we introduce the concept of active transport, we think of this. Okay? We have a protein carrier within the plasma membrane, requires the phosphorylation or dephosphorylation of ATP, the expending of ATP, to help bring things across that are going against their concentration gradient, but they are needed for other processes. And we'll see that here in a second. All right. So this is what we generally think of. Going from high to low concentration, going from low to high concentration. I don't know why I keep saying it backwards. Going against that concentration gradient. These are there to help establish equilibrium. That's why they're necessary. Help establish equilibrium. Help establish equilibrium. That's really the major function of an active transport mechanism, okay? Of a primary active transport mechanism. And here's some examples, guys. Calcium pump is an example of a primary active transport mechanism. Right? We will see it a lot here. This is probably one you are a little bit familiar with. I know in my 110 class, I go over a little bit of the phenomena associated with the physiology of a muscle contraction sequence, of a nerve impulse conduction. I don't dive into it as much as I do here, but we'll make reference to some terms here. And calcium pump is an active transport pump. It helps us to get activity to occur within the muscle cell, within the muscle fiber, or inside the synaptic knob. So that's probably one that you've seen. It's also involved with specifically pumping calcium from the cytoplasm back up into the sarcoplasmic reticulum or the cisternae. So that may be one that you talked about when we talk about muscle contraction sequence. So we'll talk about it in skeletal muscle. We'll talk about it in heart muscle, smooth muscle. So we'll be talking about calcium pumps a lot all over the place. They're also involved with the neuron as well, like I mentioned. So we'll talk about it a little bit there as well. These calcium pumps are located all over the place. Okay? They're located all over the place, and it is an example of an active transport mechanism. And see, it's closed and it requires ATP to help change the shape of that little protein carrier and get it the heck out of Dodge. Another example, if I was to ask an example of some active transport mechanisms or some primary active transport mechanisms, I guess I should say specifically, we have calcium pumps and we have this one here, sodium potassium pump. You know this one, okay? We're going to move 3 sodiums in one direction. We'll move a couple potassiums in another direction. Okay. 3 sodiums out, 2 potassiums in. Okay. Spoiler. Sodium is very high outside of the cell. Potassium is high inside. The brackets just mean concentration. High concentration inside. And those are just things that you're going to have to remember because that's going to be the driving mechanism for how everything happens that we'll be talking about over the course of the semester here. All right. So this sodium potassium pump pumps 3 of those things out, sodium, and 2 of those potassiums into the cell. This is going to be there to help maintain equilibrium, okay? Helps maintain the charge, the balance on either side of the plasma membrane. So it helps maintain equilibrium or resting membrane potential. That's all C2 and 3 are saying right there. I'm probably not going to ask. I'm not going to ask the steps. It's not going to be a short answer question at all. We're pumping 3 sodiums out and 2 potassiums in. I'm not going to ask the steps. So don't kill yourself memorizing it. All right. But there again, in the phosphorylation or dephosphorylation of ATP. We have another type of active transport mechanism that is referred to as secondary active transport. Most books always refer to it as secondary active transport. Sometimes they'll also call it coupled transport, but I haven't really seen that in new textbooks at all. It's generally just referred to as secondary active transport. So that's what I will use on the exam. Secondary active transport mechanisms are unique. Okay. It is active. Okay. So there is going to be going against the concentration gradient. Okay. But with secondary active transport, there's a second thing that gets pumped across the transporter. That second item, ion, molecule, generally goes down a concentration gradient. We see a lot of active transport mechanisms in the digestive tract. Movement of glucose sometimes requires a secondary active transport mechanism in certain situations. Okay? And we may talk about some of those later when we get into the digestive tract. Understanding there's a primary and a secondary active transport, yes. Okay. Understanding what types of secondary active transport there are, yes. That would be something that you should be able to do. There are a couple different names for this one, and please know both. And the only reason why I say know both is that I know in previous textbooks, and I think maybe in this one, I haven't gotten to one of the, or I read the whole book a little while ago, and I can't quite remember off the top of my head. But most textbooks interchange these words a lot. In one unit, they may refer to it as a co-transport mechanism, and in another portion of the book, they'll call it symport. And I don't know why. All right. These are two terms that are used interchangeably in various textbooks. So please just be comfortable with both terms. Right. I don't have a whole heck of a lot of those, but this is one where literally in the previous textbook for this class, they use symport and antiport. In the 109 class, they talk about co-transport and counter-transport. In 110, I can't remember what that textbook, I can't remember what the McKinley text used off the top of my head. But the most current ones, they use them interchangeably, so know them both, please. Co-transport or symport go in the same direction. Okay. Sodium, glucose are going in the same direction. Countertransport, they go in opposite directions. Whoops. Oppo. Directions. I don't want to spell it all out. Okay. We will see a lot of these secondary active transport mechanisms in the blood vessels, involving chloride ions with helping to make hydrochloric acid in the stomach. So we'll see a lot of these secondary active transport mechanisms in a number of locations. Okay. So we'll see these words again. Understanding co-transport or symport go the same way. You can see glucose and sodium. A lot of glucose transporters have this kind of co-transport mechanism with them. Counter-transport, they go in opposite directions. Sometimes you can have both within the plasma membrane. Okay. Sometimes you can have both within the plasma membrane. There it is. Okay. Here's an example where I have both secondary active transport and primary active transport in the same cell. You can see I have a co-transport mechanism in the apical membrane. And in the basal membrane or basolateral membrane, I have a primary active transport mechanism. Okay. In the co-transport, that symporter, sodium is going down its concentration gradient. And that's what helps us to facilitate the movement of glucose across. Sodium going from high concentration to low concentration helps us right here. We got to pump all that sodium out, right? So now I have a primary active transport mechanism. Okay, there's my sodium potassium pump. And now I have facilitated diffusion down in here. So when I talked about glut transporters and being facilitated diffusion, I'm not a total liar. Okay. We have two surfaces. We have two membranes to bring stuff across. So when we talk about what happens in the digestive system, when we talk about what happens in the circulatory system, in the urinary system, we are going to see multiple mechanisms in a cell, bringing stuff into and removing it from the cell. What are the terms that we're going to use? This is terms we're going to use. Okay, we're going to use these terms of absorption and reabsorption. Okay, but they are mechanistically active transport and secondary active transport, right? Primary active transport and secondary active transport. Absorption brings into a cell. Reabsorption is basically removal from the cell. Okay, taking it from the blood into the cell, putting it back into the blood. That's what absorption and reabsorption is. So these active transport mechanisms are going to be vitally important in terms of ions moving from the blood into tissue, back out of tissue, nutrients, metabolites, waste. If we say the word transcellular, that's the movement from the apical membrane, transporting it through the cell, and then kicking it out on the basolateral surface on the bottom of the cell. That's referred to as transcellular transport. That involves active mechanisms on both sides of the cell membrane, apical and the basolateral. And that involves mechanisms inside the cell. What mechanisms would that be? Well, I got to transport it across, right? Package it up into a vesicle, move it across. And we see a lot of that in terms of the digestive tract. All of the movement, all of the absorption from the digestive tract, from the small intestine, we absorb into those intestinal cells. And then we travel it, pass it through the cell, and then dump it back out into the blood vessels that are found traversing the small intestine. So we're going to have both mechanisms of absorption and reabsorption. So there's a lot of terminologies that we're going to be using. Just remembering that these are active transport mechanisms is just something that just needs to stick in the back of your head. Sometimes I need to move things to the sides. Para means around, side to side. Okay. So with these mechanisms here, we have to get through those junctional complexes. Remember, we have three types of junctions that hold epithelial cells together. Okay. Tight junctions, very zipper-like. Adherens junctions, and desmosomes. Okay. Little spot weld and some protein fibers to reinforce the connections between these. But it's these type of junctional complexes that are fantastic, right? Because they help maintain the integrity of the tissue. They also limit things moving in between the cells. Shockingly, we'll talk a little bit about that within the digestive tract and maybe a little bit within the urinary system as well. So these things can really limit movement side to side, but we have a lot of paracrine transport mechanisms. Okay? We have gap junctions. Gap junctions would be a way to have easy paracrine transportation. With gap junctions, what do we have? Tiny little holes open from one cell to the next. Not every cells have those. But that makes it really easy for paracellular transport. We have a number of paracrine type secretions that go and diffuse from one cell to the adjacent cell here. And we see a lot of that with how hormone signaling works. Okay? If the cell was to become infected with a virus, they release paracrine secretions that diffuse into the adjacent cells next to it and tell them to get ready to fight off a foreign invader, because I've fallen and you will soon fall if you don't make cytokines. We are going to talk about massive amounts of movement from one side of the cell, from outside of the cell to the inside of the cell. This is bulk transport. Okay? Bulk transport not only involves large substances, but a large amount of water as well. Bulk transport is what happens in the capillaries. I'll just put it up here. Capillaries. Bulk transport is how we get a lot of water and nutrients from the arterial side of the capillary bed into the tissue. And then we just have some reabsorption of some water and some nutrients on the venous side of that capillary bed. Bulk transport is endo and exocytosis. Bringing in endo, removing exo. Bulk transport, endocytosis and exocytosis is an active transport mechanism. From this picture here, it doesn't really look like it, but there is actually a whole hell of a lot of work to get that tiny little vesicle right here to fuse with that plasma membrane. To get that substances out of the cell, or heck, even to get it into a cell, requires a number of very specialized receptors. And they're called snares. Okay? And that's all you need to know it as. You don't need to know it as soluble N-ethylamine sensitive factor attachment protein receptors. You don't need to know that name. Okay? I just put it there so you know what the hell it is. Somebody always asks. That's what it means. We have a number of snare proteins. We have two major ones. We have a V snare and a T snare protein. And it's the interaction, as you can kind of see, between these proteins that allows that vesicle to go and fuse with that plasma membrane. V stands for vesicle. T stands for target. So that's where those V's and T's are coming from. But you can see on that vesicle there, it has a specific type of V snare protein. T snare protein has a specific setup on its surface. It's the intertwining of these vesicle snares and these target snares that interact with one another that get these two plasma membrane structures to fuse together here. So it's never just, oh, this vesicle just comes up there and, comes in contact with it. it does come in contact, but it comes in contact in a very specific way. The use of these snare proteins. Okay? And there are a few different ones out there in their interaction. There is some specific set of mechanisms to get these two things to kind of twirl and interact with each other so we can get this fusion to occur. But I just want you to know that's how it actually happens. Okay. We're not going to get into the whole entire mechanism of it because we really don't need to. Right? But understanding we have an interaction of V snares and T snare proteins, some specific protein markers on the surface of these two things that interact, that actually allow endocytosis and exocytosis to occur. Right? Because once this thing pinches off, it's a vesicle. Here it starts to invaginate in, okay? And here we'll have T-snare proteins here that'll start to manifest on the surface. And when this thing pinches off, those things stay there. The vesicle will have its own protein markers on its surface. Okay? So it's this interaction between these two different types of proteins that gets all this fantastic magic to happen inside of a cell. That's kind of the cool thing about this. And they can have defects. And if they have defects, then you can't have bulk transport happen as efficiently and effectively as possible.